All right. Well, thank you. I want to welcome Dr. Stratakis today to our grand rounds, and it's a personal pleasure for me to have him visit us. Dr. Stratakis received his medical training at the Kapodistrian University in Athens, Greece, and postdoctoral training at the National Institute of Child Health and Human Development, NICHD, in Bethesda. After residency and fellowships in pediatrics, pediatric endocrinology, and medical genetics at Georgetown, he went back to the NICHD, where he was scientific director, acting and appointed of the NICHD since 2009, and chief of the section of endocrinology and genetics. Dr. Stratakis has served on the editorial boards of many journals, was the deputy editor of the Journal of Clinical Endocrinology and Metabolism, and is currently the co-editor-in-chief of Hormone and Metabolic Research. Dr. Stratakis has identified genes for Carnegie complex, bilateral adrenal hyperplasias, gigantism, and other endocrine disorders, including Carney-Stratakis syndrome. The condition which he is most proud of is GPR 101 XLAG and gigantism, and also very proud that he has mentored over 200 trainees through his time at the NIH. Constantine has recently retired from the NIH and is building an exciting new research institute in Athens, Greece. And today he's going to tell us about his journey in curiosity and identifying new conditions and new diseases. So thank you, Constantine, for joining us today.
Well, thank you, Brand, for a very warm welcome and the hospitality. We had a great dinner last night, and it is a real pleasure to be here, even though I'm giving the seminar virtually. It is the first time that I'm giving pediatric grand rounds at a place, regardless of the actual virtual transmission at the end, since the pandemic. And what a amazing, horrible, whatever people say for this 18 months, almost 2 years has been. But that's what life is. And it also reminds us, all of us, two important things: how important it is to be a doctor, a pediatrician, and also the amazing contributions of science to society. And I hope this pandemic will stay in our memories forever for this too, obviously for many other things, but for these two that are relevant to what we're doing today. So I will talk to you about discovering new genetic syndromes, seeing patients, and really there's going to be a little bit of science in my talk, but most of it will be, in fact, about clinical medicine. Here's my disclosures, and I'll start with this quotation. So Claude Bernard said, he's considered the father of endocrinology in Europe anyway. A physician's subject of study is necessarily the patient, and his first field for observation is the hospital. And so even though I started my career as a basic scientist, and then I became a clinician, I understood very early that unlike the basic scientist whose field of observation is the bands, for us, it is the patient. And even if you go back to basic science, it is the patient, it is the bedside that we get all our ideas and where all our work back in the bands go back to. And then another quotation that actually Dr. Carney at the Mayo Clinic, whom I'm going to visit tomorrow with, gave me early on when I first met him. Discovery consists of seeing what everybody has seen and thinking what nobody has thought. So in other words, when you make discoveries in medicine or in science, most often, you pick up on something that somebody else has left. And I think this is an amazing saying. It really, I never forgot it throughout my career. I always said almost every day, seeing new patients at the NIH Clinical Center. And it was first said by Albert Gorky, who got the Nobel Prize in 1937, physiology and medicine. And then finally, from Sherlock Holmes. So that's a pretty famous quotation here. A Colonel Ross says, is there any point to which you would wish to draw my attention? And Sherlock Holmes responds, to the curious incident of the dog in the nighttime. And Colonel Ross says, the dog did nothing in the nighttime. And Sherlock Holmes says, that was the curious incident. So what that quotation is about recording everything that you see, whether you are working on the bench or you are with a patient on the bedside, by the bedside, you have to record everything. So not only you observe things that others may have forgotten and not see or not record. The other thing is you really have to record everything that you see. And that is, I mean, we all know it, but then you get a radiology report, right? And I'll show you one classic radiology report from one of my patients that led to a discovery where the radiologist was focusing on what the referral was for and ignored a very obvious pathology that was obvious on the MRI, just simply because he didn't follow this advice. Just report everything you see, record it, report it, write it down, because you never know when it's going to be useful. Okay, so 4 stories that led to really my life's discoveries at the NIH. First story, how the study of a rare disease, Carnegie Triad, led to discovery of a new syndrome, which actually eventually, as Brad said earlier, got my name. So what is Carnegie Triad? It was discovered by Dr. Carnegie in 1977 and describes the association of gastric stromal tumor, that's a sarcoma, pulmonary chondroma, which is a classic hamartomatous tumor, paraganglioma, pheochromocytoma, adenocortical tumors that are sometimes cortisol producing, hyperparathyroidism, thyroid tumors, and some other hamartomatous lesions. I don't know if you notice this association of paraganglioma, pheochromocytoma, and adenocortical tumors. This is the only disease in human nosology where at the same time you have an adrenomedullary and adenocortical tumor. And when I first found out about this condition, I was fascinated. I said, this is really unique. There must be something in these 2 very different cell types. Adenomedullary are neuroendocrine cells, adenocortical are mesenchymal cells that come together at some point during early development of the embryo and form the adrenal gland. How is it possible that these 2 tumors form at the same time from these 2 different cells? And which is also, the impossibility of this is obvious by the fact that there's no other disease where you see it. You either have a pheochromocytoma or you don't have, or you have an adenocortical tumor. So Dr. Carnegie is an amazing clinician. He's actually a pathologist, a trained pathologist. But what I learned from him is the 2 things I told you earlier. Look at his notes here. Very clearly written. Everybody can read it in uppercase. And he records everything he sees. It doesn't matter whether he understands what's causing it or, and that's another thing, you know, I think one of the impediments of recording everything you see is that if you don't understand something, you tend to either not note it down or you don't care about it. And that's not the attitude of a researcher. In fact, you do want to record everything you don't understand. And that's a very important thing. So this is from his notes when he saw the first patients with Carnegie Triad. So I started working with him. I came actually to Mayo Clinic, collected tissues. And by 2007, we mapped the disease to 1q, chromosome 1q. And you can see here classic fluorescent in situ analysis at the time with a probe that shows loss of the 1q in several cells from this tumor. But we actually, it's very interesting. There's no gene that is mutated on that side, at least not frequently. There is one, and that is the succinate dehydrogenase subunit C. It turns out that the most common, so there's a few mutations in SDHC, less than 10% of the time, but the most common molecular signature of Carnegie Triad that we found in my lab was an aberrant methylation of the promoter of SDHC. To this day, so we found this back in 2014, and to this day, this is a diagnostic test for Carnegie Triad. It's the only diagnostic test for Carnegie Triad that's molecular. I'll show you a histology diagnosis. So in fact, we're still working trying to find out what causes this aberrant methylation. But the disease is not inherited. It affects only females. And the defect is often a genetic mosaicism. And so we think that there's something happening in the early development of the adrenal and the other tissues that are affected by this disorder that either doesn't revert the methylation of STHC or causes the abnormal methylation of STHC in a mosaic manner and causes coronary triad. But as part of this work, we found in Dr. Carney's collection of samples and families, pedigrees that did not have any of the other tumors of the coronary triad. They only had paragangliomas and sarcomas. And here's a family. Now, consistent with my theme today, record everything you see and also investigate if you don't understand something. Here is a family that had only these two. And you can see that unless you ask for other individuals with the disease, you wouldn't pick them up. So because the disease skips generations. So we noted that in these families, unlike coronary triad that is only present in females and it's not inherited, in these families, you had both males and females affected, and the disease was inherited in an autosomal dominant manner, even though it was skipping generations. So we published this observation together with Dr. Carney, and that is what I called familial paraganglioma gastric stromal sarcoma. And of course, people could not pronounce all this, and they named it Carnegie Stratakis syndrome. And today in the literature is known as Carnegie Stratakis syndrome or Carnegie Stratakis dyad. Now, what is this disorder caused by? So unlike the methylation of STHC that is causative of coronary triad, in Carnegie Stratakis dyad, the disease is caused by classic autosomal dominant inherited mutations of the succinate dehydrogenase subunits B, C, and D. And when we first described this, and it was in a paper in New England Journal of Medicine, as you can see here, it was the first disorder where a solid tumor was caused by a mitochondrial enzyme defect. Now, succinate dehydrogenase is a unique enzyme. It serves both the Krebs cycle as well as the proton transfer chain. And so it has dual function, which is perhaps the reason why when you are a carrier of one of these inactivating mutations, you activate the hypoxia signaling pathway. But you can see here all the families we found. We found mutations in 90% of these families. So it was a classic autosomal dominant. There was no other genes causing this disease. Now, that led to a search for other genes in sarcomas that were not associated with paragangliomas. And for that, we formed what we call the NIH Pediatric and Wild Type Gastrointestinal Stromal Sarcoma Clinic, or GIST clinic. And we collaborate with major cancer centers around the country and the world. And what we found was that, in fact, 90% of the Carnegie Stratakis dyad patients had mutations in succinate dehydrogenase, but another 12 to 15% of the so-called wild-type GIST had mutations in succinate dehydrogenase. This is amazing. I mean, these are kids most of the time, or young adults who present with a gastric stromal sarcoma. They have absolutely no history of any endocrine disease or pheochromocytoma or paraganglioma, and then they present with this horrible tumor, the sarcoma, which does not respond to the usual imatinib and other tyrosine kinase inhibitors, because the mutation here profile is different. They're caused by succinate dehydrogenase mutations. And we found this because of the observations made in Carnegie Triad. We also developed here a histology test, which actually pretty simple. You may know that most gastric stromal sarcomas are bearing mutations in the KIT and the PTGR alpha genes. Well, these ones that are mutated at the succinate dehydrogenase level, they're called wild type because nobody knew what the mutations were. These ones stain negative for succinate dehydrogenase B. And it's a simple test that you can do in any histology laboratory. So today, when you have a kid or a young adult presenting with a GIST, all you have to do to find out if he will respond to imatinib or any of the sorafenib and all the TKIs. All you have to do is take the biopsy and stain it for stage B. Just B. You don't need to do C, D, or A. Just B. And because all the mutated ones will not stain positive for B, as you can see here. It's a simple test, all from that observation. Now, it turns out that these tumors, they don't stain positive for B at the histology level because they are caused by abnormal methylation, just like our carnate triad patients. These GIST patients also have abnormal methylation profile. And my former fellow, who actually did a lot of his laboratory work with me while he was doing his medical oncology fellowship at Hopkins, and he's now at Georgetown, an associate professor of oncology. He expanded this work, and we published in JAMA Oncology this paper back in 2016, showing the contribution of these pathways of methylation plus succinate dehydrogenase mutation in the ecosystem of GISTs, which is actually pretty significant. Today, we think about 1 of 7 to down to 1 in 5 or up to 1 in 5 of these GISTs can be treated with medications that will tackle these pathways rather than a TKI. So you can see how this little observation, a trip to Dr. Carnegie at the Mayo Clinic and studying his data led to significant observations that changed the lives of our patients. And also the lives of patients that I never knew they existed, right? So the GISTs, these are patients seen by oncologists, not by me, an endocrinologist. And yet I was part of a major discovery that affected the life of many oncological patients. It's just an amazing story. Our second story is the study of a single patient that led to the discovery of yet another syndrome, a syndrome that I named as the 3P, or the 3P association for pituitary adenomas, paragangliomas, and pheochromocytomas. Now, how did this observation came about? Well, I was always interested in diseases of multiple endocrine tumors. And I knew that when I was looking at the paragangliomas with Dr. Carney, that there were a few patients that had some other endocrine tumors in addition to the paragangliomas and pheochromocytomas. And I had seen in the literature papers like this one, as far back as 1976, reporting this weird associations like a prolactinoma with a paraganglioma, or a pheochromocytoma with a corticotropinoma, a Cushing's disease. So it's strange stuff, pheochromocytoma and Cushing disease from the pituitary. So anyway, at the same time, in my cohort that I was studying for the pituitary tumors, this 37-year-old man presented with signs and symptoms of acromegaly and unilateral hemianopsia. It was obvious he had a large pituitary tumor, but he also had a 3-year history of hypertension, which obviously every endocrinologist who saw him and realized he had acromegaly attributed to acromegaly. We all know acromegaly causes hypertension. And so he was referred to a radiologist. Here's what I was telling you earlier. The radiologist knew that he was seeing a patient with acromegaly, the MRI of a patient with acromegaly, and boom, he focused on the MRI on this very large pituitary tumor, very obvious. Right here, you can see the pituitary tumor occupying the sella, extending above the sella, but not pushing the stalk upwards, which is intact. You can see the tumor here too. And what these tumors often do, they push the pituitary tissue around, and you can see with gadolinium enhancement, the enhanced rim of the pituitary, whereas the tumor doesn't pick up gadolinium. Okay, now I don't know if you can see what I saw when I saw this MRI, which the radiologist actually missed. So looking at this MRI, I said, what is this? And the fellow sitting next to me said, oh, it's, he has acromegaly, he has a thick neck, the patient. I said, what are you talking about? This is not outside the neck, this is inside the neck. There's something there, right? Here it is. And lo and behold, we did what we were supposed to do, got a CT and then an angiogram to try to see what these were. And they were highly vascular tumors, as you can see here in this angiogram, which of course told us that this must be paragangliomas, carotid body tumors, right? And so, if you make a diagnosis of a carotid body tumor, then you have to do an FDG PET, the usual MIBG. And of course, here's the tumors of the neck. They were positive on FDG PET and FDOBA and MIBG, but MIBG very clearly shows two pheochromocytomas, which were also picked up by the Octoscan and the FDG PET. So here's a patient who had acromegaly, yes, had this large pituitary tumor, but he also has multiple paragangliomas and bilateral pheochromocytomas. And then I asked the patient about his family. You can see here he is. His sister also has severe hypertension, and all the people there with a dot are hypertensives. We were able to get the sister and screen her, and she also had multiple paragangliomas. So what did this patient have? Well, this was in the middle of my study with paragangliomas, and so quickly screened for succinate dehydrogenase mutations. And the patient ended up having a mutation, a protein truncating mutation in succinate dehydrogenase subunit D. And so then we took the pituitary tissue out and we did classic cancer genetics, trying to see whether this D subunit had lost the normal allele. And you can see the normal allele was lost. Western blot showed loss of SDHD expression, and then loss of SDHD expression also at the pituitary level. So here's a normal pituitary with plenty of SDHD, normal pituitary with plenty of SDHD, an abnormal pituitary from another patient with plenty of SDHD. And here is the pituitary tumor of this patient, the growth hormone producing tumor, doesn't express any SDHD. So we published this case and we said, okay, well, there's so many other cases in the literature of pituitary tumors and paragangliomas and pheochromocytomas. Maybe this is the beginning of something else. And in fact, we reported in this paper how many other cases exist all the way back to 1952. And so we wrote a hypothetical review of all these cases, but that led to investigating all cohorts of pituitary tumors we can get our hands on in the United States, more than 200 pituitary tumors. And we found several cases, in fact, of succinate dehydrogenase mutations that were present in patients that had pituitary tumors, and they themselves or their family members had pheochromocytoma or paraganglioma. We named this condition 3P, and of course, it's always nice to confirm your data with somebody else or with an animal model. And here's the animal model. We SDHB heterozygote mouse that gets prolactin hyperplasia, and they also get this prolactin producing cells that are hyperplastic in the mouse. They get mitochondrial abnormalities, of course, due to the SDHB defect. But the interesting thing is that we see the same mitochondrial abnormalities in the patients with the SDHD tumors. And we confirmed it in another cohort in England with Marta Carbonets. And of course, there have been several cases since our publication of patients with succinate dehydrogenase mutations, the 3PAS syndrome and pituitary tumors. And interestingly, there's also now, this is the first case of pituitary cancer due to an SDHB mutation. And there's another case I know of that has the same thing. So, a simple observation from a simple patient, an MRI that had been misread by the radiologist led to the description of a new disease, really fascinating story. My third story is, well, I have many favorite things that happened to me in my career, but this is really my favorite disease, as Brad said. I started collecting kids that were overgrowing back in 1998, 2 years after I finished my fellowship. And while I was completing my Carnegie complex project, I got interested in gigantism. And what I noticed was something that people hadn't seen before, which is, as I said before, right? So something that I observed that people had ignored. Not that it wasn't there, it was there. But what is that? Is that for a certain group of giants, for acromegalic giants, there is this subgroup of patients that got their overgrowth in the first year of life. Like this girl here from Vietnam. You can see that she tripled her size within the first year. Very unusual, very, very unusual. And when I looked at growth charts that were available of giants, you could see that there were essentially two groups of giants in pediatric growth hormone overproduction. Those that would overgrow the first year of life, like this, and those that didn't, that overgrew later on, after the age of 7 or 8. So I studied at the same time I saw this Vietnamese girl, a family from New Zealand. Here's this boy, he's 4 years old, and look at the height, and this is his father who is unaffected. And this boy is 2 years old, and look at his growth. Now, they both got their disease from their mom, who had complete hypophyseectomy for the same reason when she was 18. She's obviously normal height today and doesn't have any signs of gigantism because she was treated when she was young. When all these patients were operated, the mom, the two boys, and so the mom was operated in New Zealand, the two boys were operated at the NIH, we noted that their pituitaries were mostly hypoplastic. Mom had a tumor, but she was operated when she was 16. The boys had a simple hyperplasia, no tumor. And so the boys had also this characteristic growth of over the first year of life, tripling their size. And we looked in the literature, and Ora Peskovitz, Ora Peskovitz and Erika Oxter had published that paper in JCNM, a classic, very nice review. I think the best until we started studying the thing. 100 cases of pituitary gigantism, which I think Erika wrote or collected over the time she was a fellow. So in that review, it's pointed out that many of the very overgrown giants, including the tallest man we know, Robert Wadlow, which Brad reminded me is from Illinois and not from Ohio. So the Alton giant from Alton, Illinois, not Alton, Ohio. This man too grew dramatically the first years of life, and of course he ended up being the tallest man. So all these patients were men, and nothing was pointed out in this paper about possible X-linked disease, because obviously there's also a few women, but it was weird that the ratio was amazing for men. And then there is this old publication from actually the guy who described acromegaly, Pierre Marie in France, who described the Hugo brothers. And the Hugo brothers, of course, there were two boys that overgrew the same way the Alton giant overgrew. And so I was convinced by the time I saw the Vietnamese girl and the two boys from New Zealand that I was dealing with a separate form of gigantism that was affecting these kids early in life, like the first year of life in infancy, first 2-3 years of life. And second, that it was likely to be X-linked. There was something about the sex, distribution distortion that made it likely that this was an X-linked disorder. But I started sequencing the thing, the whole genome of all these patients, about 26 patients, and I found nothing. I mean, in fact, my first lead was wrong, and I got frustrated until I started looking at something else. In my whole genome data, and remember, this was now 2014, so it's already 7 years ago. The technology wasn't as good as it is today. I started noticing in some of our whole genome data an abnormal signal on the X. And lo and behold, all these patients had what is shown here in red, this abnormal amplification signal on the long arm of the X. And I said, my gosh, this can't be, statistically, it couldn't be by by chance. It just couldn't be by chance. 26 patients, all 26 have this amplification. Now, what made it difficult was that it wasn't exactly the same, as you can see in this graph. The borders of the region were different. So this was a de novo defect in these families, inherited from either from the mother to both males and females, and the females could transmit it, but the males were more severely affected. And then we noted, though, that all of these patients had one region in common that included primarily an unknown G protein coupled receptor, GPR101, that is expressed in the hypothalamus. And at that time, it wasn't known whether it was expressed in the growth hormone producing cells. And it turns out it's also expressed in the pituitary and the growth hormone producing cells, but only during fetal life and early toddlerhood. So somehow, after the age of 3 or 4, GPR101 in normal pituitary stops being expressed and gets reactivated its expression during puberty. Fascinating story. It's a fascinating gene. We're still working on it. And that led to this paper and the naming of the disease. We called it XLAG. I like naming diseases. And really how this came about was one day sitting over my postdoc, and as we were writing the paper, I said, how should we name this? It's X-linked. These people wear extra large, right? So let's call it XLAG, X-linked acrogigantism. But really, how I came about with this name was from extra large and the X-linked. So extra large, X-linked acrogigantism. Today, we have a fish model for that. We published in Human Molecular Genetics this year. We knock out GPR101, and the fish grows short. You can see here that this GPR1 knockout fish, this is the green, the knockout, is much shorter than the heterozygote, which is almost like the normal. And here's the normal. And they're deficient in weight as well, and the BMI. So, a simple observation, the growth chart, led to this discovery, which I'm so excited about. Clearly my most favorite discovery because of the many turns and the many years it took to find this disease. And my final story is, of course, on Cushing's syndrome, adrenal Cushing's syndrome. I love Cushing's syndrome, and today I'm going to present to you my work on Cushing disease at the residence or the sections session. So I will only talk about adrenal Cushing's here. So what is the lesson from this fourth story? When the data don't fit, don't give up. How believing a mother that was really very frustrated with the medical system led to the recognition of a new form of adrenal hypoplasia that affects mostly toddlers and young children. And something that I often say to the fellows, I think it's so true in medicine, it's so true in life. Truth is the daughter of time. For people that are history buffs, this is, of course, the Greeks said it first, of course, but it's better known with its Latin inscription, Veritas Filia Temporis, truth is the daughter of time. And it was the motto of Queen Mary Stuart, for those of you that are history buffs. So it's in the coins of Mary Stuart, you can see Veritas Filia Temporis. Such an amazing saying. People have to understand that truth always reveals itself. It may take time, that's true, but you have to be patient. Here is the patient that it proves this Latin inscription so true. This patient was born with Cushing's, as is obvious here. So he had Cushing's all the way to the end of the first month of life. And then by the time she was 2-3 months old, you can see Cushing's has melted away. By the time she's a year and a half or so, she's had some signs of Cushing's. Clearly Cushingite by the time she's 2 and 2 and 4. Look at her by the time she's 2 and 10. Cushing's has gone away. Now Cushing's comes back just around the time she turns 3. And 3 years and 1 month, no Cushing's. So this is what we call periodic cyclical Cushing. So mom was almost prosecuted for Munchausen by proxy. And so they wrote to me from Seattle Children's, and I flew the girl over to the NIH, and back and forth over 3 months, we were able to document the same thing, but this time measuring cortisol and knowing, and videotaping and everything, knowing that this had nothing to do with mom. And you can see here with Cushing's in December of 2002, no Cushing's in February of 2003, fulminant Cushing's in April of 2003. And in between, variation in the cortisol levels from like 10 milligrams over 24 hours, minimal excretion here, to over 1,500 milligrams over 24 hours here. So something like 10 times more than 10 times, 20 times the upper normal. Now, the CT of the adrenal shows what the radiologists always call normal adrenals. Actually, if you look carefully, they're not entirely normal. They have small little dots showing that there's small nodules there, micronodules. So this is a form of micronodular hyperplasia. And how do you prove that? Even in times when the cortisol levels are normal, all you have to do is give dexamethasone. So in 1998, I described this diagnostic test, which is still in use today. You give the patient over 6 days, low dose first, then high dose. This is a repetition of the classic Little's test, but this time to stimulate rather than suppress cortisol secretion from the adrenal. And I've published the guidelines for how to interpret the test. Basically, you need a more than 50% increase of either UFC or 17-hydroxysteroids. Of course, people don't measure 17-hydroxysteroids today, but urinary free cortisol, therefore, by day 6. So the 2 basic tests days, you measure the baseline and you compare that at the end of the high dose. And if the UFC rises above 50%, no matter what the baseline was, this is pretty diagnostic of micronodular hyperplasia. And this is how the adrenals look here in this disease. You can see they're hyperplastic. They have nodules growing outwards, which is only seen in cancer. This is not cancer. These are fetal cells that grow outwards, as we have shown in mouse studies. There are several patients with this disease. To this day, well, I'm working on the genes, actually. So there's a few genetic defects we have found. So I hope I will publish them in the next year or two. It's a bilateral adrenal hyperplasia. This is one of my other patients from Pakistan. He did very well, but you can see the adrenal, multiple little nodules. And this is different than Carnegie complex. This is not PPNAD. You can see the adrenals here have a little bit of pigment, but they don't have the dark pigment of primary pigmented nodular adrenocortical disease. And of course, these patients don't have PRKN alpha mutations. What do they have in terms of genes? Well, we published few of these patients have mutations in phosphodiesterase 8B, and other patients have mutations in phosphodiesterase 11A. And it makes sense because these phosphodiesterase defects regulate cyclic AMP, which is defective also in Carnegie complex and in McCune-Albright. But the majority of the patients have other defects that, as I said before, I'm working on. Now, yet another observation from the same cohort of patients led to another big discovery. Just 5 of these patients that I had a very peculiar form of micronodular adrenocortical hyperplasia that was in between PPNAD and isolated micronodular disease. So IMAD and PPNAD. What I mean by in between? Well, some patients had pigment, some didn't. The tumors were somewhat larger than micronodular. And I had learned my lesson with the acromegaly, where I learned to look at dosage of DNA rather than simple mutations. And to make the long story short, I discovered that these 5 patients with Cushing's have different amplification signs of chromosome 19p. And what gene is on chromosome 19p? Boom. It was the catalytic subunit of protein kinase A. So amazing. This is right in the heart of the cyclic AMP signaling pathway. So it made sense for me to look and see if PRKCA was involved. And PRKC was involved. In fact, the larger the amplification, the more severe the disease. And that led to a collaboration with a group in Germany that was actually working on the same subject but this time on PRKC alpha mutations in sporadic adenocortical tumors. And together, we described this mutation, the L205R, which is a hotspot mutation for the catalytic subunit of PKA, the same gene, that is mutated now, we know, in about half of all cortisol-producing tumors. And this has nothing to do with germline genetics. This is a somatic defect now. We described this thing in New England Journal of Medicine, and it's one of my most highly cited papers today because of the impact. But it all started with patients with macronutrient disease and this select group that just didn't fit with the others. And looking at their genome data led to discovering the PRKC alpha. Now, at the same time, we were also looking at other forms of hyperplasia. Remember, I just told you that these 5 patients were sort of in between. And what I meant in between IMAD and PPNAD, but there's also this other disease called macronodular adrenocortical hyperplasia. And I mentioned that some of the PRKC alpha patients had large tumors. Well, I had a whole other group of patients that all they had was large adrenocortical tumors producing cortisol. And a group in France and in Canada, André Lacroix and others, published back in the mid-90s that this patient had abnormal expression of G protein coupled receptors. So somehow this macronodular disease was also linked to the cyclic MP signaling pathway. So I collected these patients and found some mutations. In fact, some had mutations in MEN1. Others had a mutation in the gene of the cycle of Krebs, fumarate hydratase. One patient had familial polyposis coli, and one patient had McCune-Albright. But the majority of the patients didn't have mutations in non-genes. And now today we know that some patients with McCune-Albright have macronodular hyperplasia. We know that patients with familial polyposis coli have macronodular hyperplasia. And we know that 35% of all MEN1 patients have bilateral adrenal tumors. So this was the first observation published back in 2009, but today it's a well-known fact. But what about the other patients with macronodular hyperplasia that don't have these other tumors? Well, we put them together and we did a genome-wide screen, and we found that they had a number of chromosomal abnormalities, which is abnormal. It's also abnormal for a benign tumor. Now, these tumors never become cancer. So why would they have so many genetic changes? That's not clear to me even today. But the important thing was that in this genome-wide screen, we found that one chromosome, chromosome 16, was most frequently involved. So we sequenced this chromosome up and down, and found out that all the patients that had this amplification signal, this loss signal on chromosome 16, had mutations in a gene that nothing was known about before we started studying on it, ARMC5. So ARMC5 for armadillo-like repeats. So this gene is very much like beta-catenin, which also contains armadillo repeats, which is on 16p on the short arm of chromosome 16. And all these patients had inactivating mutations in chromosome on ARMC5 on chromosome 16p. What's interesting is that not only these patients had a germline inactivating mutation, but each one of the nodules, so you can see here multiple nodules, right? Each one of the nodules had an additional mutation on the other allele, completely inactivating the gene. And some patients had as many mutations as 20. I mean, unbelievable. So what you see here in this 60-plus-year-old lady with MMAD, with macronodular disease, is that she was born with this germline mutation in black, but then every single one of these nodules had another mutation in the other allele, completely inactivating the gene. So extensive genetic variants. Now, we have described several families with this today. And the important thing is, why I'm including it in a pediatric talk, is that many of these families have their patients that are carriers, starting on with obesity and hypertension at about the age of 16, 17. So I know it's rare, but for those of you that run obesity clinics, remember that once in a while, I mean, very rarely, I agree, but The possibilities out there that in your patients, 16-year-old with obesity, hypertension, and stretch marks, that could be, just could be, a patient out there with macronutrient hyperplasia. Now, how you pick them up? Yeah, well, that's where genetics plays a role, and we're not there yet to suggest massive screening for MC5. But just remember that possibility exists. So I'll finish up here and leaving some time for questions. I think what I took you through, and here's the NIH building where I spent 28 years of my life. And it was 28 good, very good years. The most beautiful thing we can experience is the mysterious. It is the source of all true art and all science. He to whom this emotion is a stranger, he who cannot longer pause to wonder and stand wrapped in awe, is as good as dead. His eyes are closed. And that's by Albert Einstein. And I was, from very early on, enticed by the unknown. I mean, I always kept asking questions. Why is this happening? And that is what kept me going, and eventually led to this discovery. So if I have a message today to the residents and fellows and students who may be listening to me, if you are interested in science, follow your heart, record everything you see, and look at things that others may ignore. You never know where they're going to end up these discoveries. I was reading the citations for the Nobel Prizes this year, and every single one so far, in science, in chemistry, in physics, and in medicine, were observations that were made by people that looked at things and did not ignore them. Others had missed it, and it's just and then they followed their heart, just like Albert Einstein says. So I'll finish up here with credit to, as Brad said, I was very fortunate at the NIH, so in my 28 years, to train more than 200 people, some of them in extraordinary positions today. This is the latest picture of my lab before I announced that I was intending to retire. I retired this year and took another opportunity to build a new building, a new institute dedicated to personalized medicine, which I really believe in. I think we live in an amazing revolution of this marriage between technology and medicine that will only lead to great things. But it doesn't mean we shouldn't be good clinicians. And I hope that I pass this message to you today, just like I've passed it to all my fellows and trainees. Be a good doc. And no matter what the technology is, be a good person, be a good doctor, and that will make you a great scientist. Here's celebrating our lab. The year 2014-16, we had 5 papers in New England Journal of Medicine describe 5 different diseases and mutations, and it's a feat that's unlikely that I will ever repeat. It's due to NIH funding, of course, and the many patients that entrusted us with the care, but of course all the hard work of the people that you see here. Thank you so much for the opportunity to present to you today.
Constantine, that was an amazing and inspiring presentation. So thank you very much for joining us and telling us about this journey. So I have a few questions and a few comments. First, I forgot to tell everyone that you not only had published 700 or more papers, but you had actually survived 6 weeks in Rochester, Minnesota in February and January and February 1994, which around here gives a lot of credit. So he was telling me that when it got above freezing. Being from Greece, he was shocked that it was so joyful and nice out that you wanted to celebrate just rising above the freezing mark. Seriously though, question that I have goes back to the GPR 101 expression that you talked about. Whereas in normal individuals, it's present and expressed till age 3, and then suppressed until puberty and comes back. And I think that when I think about how kids grow and how the big growth spurt to age 3, and then the pubertal growth spurt happens, that sort of mechanism, as well as same process for puberty, what is allowing those genes to, or causing those genes to be suppressed? Is it an active process, or is it a silencing and then a reactivation later? Is there any information for GPR 101 or others like it?
Well, great question. And let me just add to what I said about the expression pattern of GPR 101 is that not only gets reactivated at puberty time, just about when we experience peak growth, but also that reactivation only lasts for about 3 to 5 years, and then it gets silenced again. So it's a highly regulated, it adds to what you asked, it's a highly regulated gene. So what is responsible for that? Funny you're asking. We have a paper that I'm working on that we're going to submit to the American Journal of Human Genetics, hopefully in the next week or two. And we were able in the last 2-3 years to identify a region in upstream of the promoter of the GPR 101 that has one of these, I hate to be a molecular biologist geneticist here, but what's called a TAD region. So this is one of these regions basically where enhancers and suppressors come together and regulate a highly regulated gene expression. There's many of those in the genome, and typically they don't control just one gene, they control many in the neighborhood. They are developmentally regulated. And so we discovered a TAD region upstream of the start site for GPR 101. And we think this is the region where this regulation takes place. What we don't know, although we know that the transcription factors that regulate this region, what we don't know, and that's the essence of this paper, is a description, again, observation paper. We describe the sequence, we describe the TAD structure, we show what molecules, known transcription factors, bind there. But we didn't go the next step, which I think is the next step in this work, which is, okay, now you know the transcription factors, you know the region. Which one of these transcription factors, enhancers and suppressors, is doing what we just discussed? And that's unknown. There's several candidates. In fact, interestingly, MEN1, menin, is one of the genes that binds there. And so I don't think it's menin that does what we just discussed. It's also possible it's not a single transcription factor, right? It's just an epigenetical phenomenon that allows the binding of factors. This remains to be discovered in proper experiments. These experiments are hard to do, primarily because of something I faced during all this work, primarily because of the absence of a reliable human pituitary cell line. And I don't think we will ever get a human pituitary cell line. I think, though, as we discussed yesterday at dinner, that the era of cell lines, and probably of animal models, is over, or close to be over for most things anyway. And looking forward to getting a somatotroph organoid where we'll be able to do these experiments in the lab. We're not there yet. There's no somatotroph organoid I know of, but that's the proper experiment, right? So you will hopefully grow this organoid that will secrete growth hormone in the lab. You will perfuse it with various transcription factors that bind to the TAD region and suppressors, and identify what causes the silencing or the expression of GPR101. That's how you should test in vitro what we have found from the in silico analysis and the few in vitro experiments to demonstrate the binding of these transcriptional factors. Because there's no, the one cell line we use for these studies is the rat GH3 cells. They're not too bad, but they're rat.
Yeah, so I have one last short question before our time is up here. And you mentioned the observation and the importance of observation. Can you, or do you foresee taking the big data of electronic medical records and being able to crunch some of those associations to help you identify new diseases? Or do you think the EMR is just not capturing enough information because no one's putting it in anymore?
Well, very good point. Let me just start from the last observation. I always say in silico research, garbage in, garbage out, right? So it's extraordinarily important for people to put the right observations, the right data, not the wrong data or no data in EMR. Okay. Having said that, the beauty of large data sets is that, yes, the occasional errors or the lack of recording will create a noise. But you have such a large volume that you can overcome what you lose from the noise. Now, when it comes to rare associations, a single error in an EMR can lead to what I said before, garbage observations. But for common diseases, you're able to overcome the noise. So part of my moving out of NIH in this next career is, I really want to study. I will finish the work on the diseases I work now on, but I'm not going to take a new project on a rare disease and try to use the available data. Unfortunately, Greece has a good national health system to do that. So the available data to find associations for common diseases that I know something about. Obviously, I don't know a lot about, pancreatitis, but I know quite a bit about metabolic syndrome and low renin hypertension, which are all extraordinarily important diseases in our aging population. And so I plan to use my knowledge of these pathways that I talked about today to find these rare associations. But again, the principles are the same. Be a good doc, try to get good data as much as you can from clinical phenotyping, and then never throw away any observations. Great. Be critical of what you keep, right?
Yeah.
But you don't throw it away. It doesn't mean you believe it. I mean, that's what I mean by being critical.
Yeah.
You always have to be critical of your data.
I think we're at time. And again, I want to thank you for an inspirational talk. Emily, do you have any last housekeeping items